Okay. So we're almost done with lipid lecture 2. So any questions on this that we went through before? Any of the steps? Digestion, absorption. Remember, we're simplifying this. And we talked about lipoprotein metabolism. And my suggestion was, break this into 3 different parts. Learn the chylomicrons, which is just basically starting in the intestinal cells, delivering dietary triglyceride to tissues, and then chylomicron remnants going to the liver. So you want to add in details about apolipoproteins and the enzyme process. And then VLDL and LDL go together. And we'll talk about that today. And then the last one was HDL, which we'll talk about in a few minutes. So one of the points that we're going to focus on today is this step where the LDL are taken up by cells. And we talked last time about that hypercholesterolemia, about how that was the kind of the impetus for figuring out LDL metabolism. So we'll talk about that in a minute. The summary is here. Right? So I have notes for this that I can post if you want on the step-by-step on this. And I can put those on there, and then there's I'll just post my notes on PolyLearn if you want. So you can take some brief notes here and then check your notes against mine when I post them. But we said before that this is LDL receptor mediated endocytosis. So in other words, there is a receptor specific for LDL. And those receptors are on almost every cell in the body. And the reason is because the liver makes a lot of cholesterol, and then it can provide cholesterol to extrahepatic tissues. Right? So that's kind of what we're looking at. We're looking at how cells take up cholesterol. And that's mostly in LDL. That's how they get their cholesterol. All right, so this is a common process that you'll talk about like next quarter with B12, with iron, with some of the other nutrients. Cells have ways of taking specific nutrients out of the blood. In this case, we're not talking about a nutrient, but we're talking about something that cells need. And so this is cholesterol. So LDL cholesterol, absolutely essential. Although sometimes it becomes problematic. So we'll talk about that in a second. All right, so here we go. So we said plasma membrane of almost all cells in the body. And here's where we start. There's 10 steps, but we'll make this shorter. So here's LDL, and this apolipoprotein right here is ApoB100. It doesn't look like that, but that looks good in the diagram. So here's ApoB100 protein. Here's cholesterol esters that are inside the LDL, and then remember it's got phospholipids on the outside. So in step 1, ApoB100 binds to LDL receptors on the cell membrane through protein-protein interactions. ApoB100 binds LDL receptors on the cell surface through protein-protein interactions. All right? Don't worry about this clathrin. That's an intracellular protein that allows the cell to engulf the particles, but don't worry about naming that. Okay, so now if you look at what happens, this cell is going to bind this. Now, why do I say protein-protein interactions? It's structural interactions. Right? So what affects the structure of a protein? Amino acids, their interactions. What affects the amino acid sequence? The gene, the genetics. So this is one of the reasons why we say, you know, genetics can actually affect LDL cholesterol levels. And I said familial hypercholesterolemia is a genetic conditions where some people have significantly elevated blood cholesterol. So now imagine this. What if the ApoB100 gene is altered in someone and it doesn't bind the LDL receptor very well? Then these particles can stay in the blood longer. The LDL receptor gene can also be altered, and its structure might decrease the affinity for B100. And that can be problematic because now the LDL wouldn't bind as well and it would stay in the blood. Okay, so those are two genes. So if I ask you to talk about genetics about LDL cholesterol, then those are two genes that you can mention. Remember, alterations in either one of those genes can cause compromised protein interactions. LDL receptor and ApoB100. As a matter of fact, you know, most of the time when you go in and you do a lipid panel, if any of you guys have ever done that, they'll do your total cholesterol, they'll do your HDL, your LDL. Sometimes in certain labs, if they want to be more specific, they'll look at ApoB100 in the blood. And there's a stronger association between that and LDL cholesterol than B100. So sometimes it's a more accurate way of quantifying that. Okay? So that was step one, right? So in step two, the cell surface engulfs or endocytoses the LDL. And an endosome is formed. Right? That's an enclosed structure. So you can call it a vesicle or an endosome. But look what's inside the LDL. So the cell basically engulfs the LDL particles that were trapped when they bound to the LDL receptor. Okay? The endosome pH is decreased to approximately 5. With the addition of protons. So there's proton pumps in this membrane that pump protons into this, and how the pH changes. So it goes from pH 7 to pH 5. So an acidic endosome is formed. And what does that do? That alters the interaction, or let's say that separates the LDL receptor from B100. The decreased pH separates the LDL receptor from B100. The LDL receptors are separated and recycled to the plasma membrane. Sure. The receptors are separated and recycled to the plasma membrane. So in other words, they're reused. All right? They sit on the cell surface, bind LDL, they're involved in endocytosis. They're separated using the change in acidity, and now they go back to the membrane and do that again. They can cycle through that. Look at the other one now. The vesicle containing LDL fuse with lysosomes, or you could say, let's say merge, it's better. The LDL containing vesicles merge with lysosomes. The ApoB100 is hydrolyzed to amino acids. Cholesterol esters are converted to cholesterol and fatty acids. Okay, so the cholesterol can be used by the cell as cholesterol, or it can be stored as cholesterol esters. So those are the basic steps. So it gets a lot more interesting when we figure out how this is regulated. So let's say a cell needs cholesterol. And we don't have time, unfortunately, to go into this class, but the cells actually have proteins that sense how much cholesterol is in a cell. And some of these proteins basically will sense how much cholesterol is in cell membranes in the cell. And then they are kind of sensors for what's called cellular cholesterol status. Those proteins can then respond by increasing cholesterol uptake. So how would that happen? What would you modify if that cell needs more cholesterol? More receptors. So in your notes, put regulation of LDL receptor levels. Regulation of LDL receptor levels. So in the first point, let's say intracellular cholesterol levels. Intracellular cholesterol levels modulate LDL receptor gene transcription. Period. High intracellular cholesterol decreases LDL receptor gene expression. Sure. High intracellular cholesterol decreases LDL receptor gene expression. Does that make sense? And the opposite. If it's low intracellular cholesterol, what happens? LDL receptor gene expression goes up. And it's through a transcriptional mechanism where a protein basically moves through the cell, goes into the nucleus, and activates gene expression through what's called a sterol receptor element. And we won't go into all that. But that's what's happening here. So here's a really important point. That's not regulated by blood cholesterol, so don't say that. It's intracellular cholesterol that's driving this. That's really important. It's intracellular cholesterol that regulates LDL receptor levels. Okay? So for example, let's say a person has low intracellular cholesterol values, and yet the LDL receptor gene has been modified in that person so it doesn't respond to this. So now what happens? Their cells are low in cholesterol, but they don't pull more cholesterol out of the blood, which is typically what happens. So what happens to those individuals? Cholesterol stays high. And that's what can happen in some of these genetic scenarios. And obviously there's different levels of modulation and effects depending on the genetics. But that's definitely one of the things. And that's what was happening in those kids that I mentioned before. They weren't pulling cholesterol from the blood. So cholesterol wasn't going into cells. It was just kind of ending up in interstitial areas and tissues. And that's when it was really problematic. And it wasn't down-regulating some of the other things that it should. Okay? So with that said, now let's take something that you guys learned, for example, in the fiber lecture. We just said LDL receptors are pretty much everywhere, right? When we went through this, I said once we have LDL, they can either be taken up by the liver or they can be taken up by extrahepatic tissue. And what percentage did we have here going to the liver? Close. 75%. 50% of VLDL become LDL. 50% go to the liver. But this percentage right here, LDL being cleared by the liver, about 75%. And in some places they refer to that as inefficient absorption. Okay? And 25 goes here. So here's what you can do. We talked about this before. We talked about recycling of bile acids. Remember that? And then we said later on, well, what happens if you take cholestyramine or fiber? Some fiber basically compromises bile acid recycling. And we said if bile acids are not recycling to the liver, what does the liver do? Takes cholesterol, converts it to bile acids. What would be the net effect of that on liver cell cholesterol concentrations? If bile acids are not recycling back to the liver from the gut, and the liver is taking cholesterol and converting it to bile acids, that's going to decrease intracellular cholesterol concentrations, right? So what does the liver do to its LDL receptors? Makes more. I think about a third of us are on the bus here. Let's look at this. So here's the liver. I think someone's bringing in their markers that are almost out of ink, so I'm going to leave them in that class. So this is the liver. Right, and we said, for example, if this is the gut, we said, let's say bile acids are here, and they're going down the gut, and some of those bile acids are doing this, they're leaving the gut, they're going in the portal vein, and they're binding albumin, and they're coming back here. And then we said, if we stop this recycling, then this bile acid is not arriving in the liver, so that's not getting there. So the liver does this, converts cholesterol to bile acids, and then the bile acids are exported from the liver into the bile. So here's an example of this, right? So now what happens? That would cause cholesterol intracellular concentrations to go down. So what does the liver do? Intracellular cholesterol concentrations are down, so the liver can do this. What does that shape look like? This, right? These are LDL receptors. They would go up. Why? Because intracellular cholesterol is what's driving LDL receptor numbers. So if intracellular cholesterol goes down, LDL receptors go up. So what happens? More LDL are cleared from the blood. So what happens to blood LDL cholesterol values? They go down. the higher number of LDL receptors causes more LDL cholesterol to be taken from the blood. So that causes blood cholesterol, blood LDL concentrations go down because they're being cleared by the liver. And I said when we first started talking about this, I said the LDL, the liver is the most important tissue for cholesterol metabolism. And here's another reason. Pretty much in here, right? If you're not sure about the answer, what's the most important? Say the liver. Right? The most important, the liver.
So the material that makes the LDL receptors, is that coming from inside the liver, or okay?
Yeah.
Are they not being like shut off and on, or they're just being formed and broken apart again?
Transcriptional regulation. Right, and how do they get there? Secretory pathway. That's the other thing you say almost every time, right? The liver, secretory pathway, glucose. You got most things covered in here. Okay? Right? It's that transcriptional mechanism that now the secretory pathways decrease because you've made less protein, less receptors to get to the surface. In this case, it's cholesterol going down that stimulates LDL receptor gene expression. And so you end up with more receptors there. And the opposite is true as well, but most of the time we're concerned with how do we decrease a person's LDL cholesterol. That's where humans have most of their cholesterol in the blood. That tends to be the cholesterol that's most problematic. That's the one that can be involved in atherosclerosis. So there's a couple things that we can do. One is fiber. Right? Let's say we have dietary fiber here. That dietary fiber can bind bile acids and decrease recycling. What else can fiber bind? Cholesterol. If it's binding that cholesterol and the cholesterol is excreted, then you're losing more from the body, and that also tends to stimulate this. One of the most effective ways, though, is this. And some of you might know people that are on lipid-lowering medications. And you guys watch different, I don't even know if anybody watches TV anymore, but let's say stuff on the internet, right? And you see these ads for, like I used to watch some golf, not much, but stuff like that. And the golf commercials are investment companies. Cars, mostly Buick, Mercedes, lipid-lowering medications, right? They're looking at a specific demographic and some other stuff that I won't go into. But that's kind of that's a huge part of the drug industry. Probably one of the most successful drugs ever are the statins. Crestor, Lipitor, Lovastatin, those drugs, they decrease cholesterol synthesis in the liver. So what does that do? They're using this mechanism. That's why I said it was Nobel Prize material when they figured this out, because now they understand how the receptors are regulated. So now what they can do is then they came out with drugs that decrease hepatic cholesterol synthesis. And so what happens? Cholesterol concentration goes down. LDL is pulled from the blood. Are they significant? I gave you this number before. Sometimes they can decrease LDL cholesterol 40 to 70%. It's a huge drop. Yeah. So it would benefit some people to be on them. And I would just say, I think we all know that, but there's a lot of people that shouldn't be on them that are. Sometimes it's the first thing. Oh, your cholesterol is a little low. Have you ever thought about being on statins? No, it's never been low. It's never been high. And sometimes it's the first thing when we know it should be. About your lifestyle, can we change your lifestyle? Can we change your nutritional intake? Can we change some of these other things before we put you on a medication? It's like a few years ago I went in and had my blood pressure was a little bit high at one place. Right away the physician, maybe have you thought about hypertensive drugs? I'm like, it's never been high. A month later, I go into the dentist. She says, is your blood pressure always that low? It moves around. Right? So, but that's very common, and they can be very effective. Obviously, there's some side effects. 329 Lab will go into some of the side effects and more about how they work, but that's kind of what's going on. So we can do things in the diet that actually change this, other things as well. And remember, insulin is also stimulating this. Insulin stimulates cholesterol synthesis. So we want to make sure that we're kind of controlling insulin release throughout the day. Because that can also affect cholesterol levels. Okay, you'll see some of this in the notes. So I think I covered everything there. Any other questions on this? Yes. I don't know. I don't know if it's why they fast. It's a potential benefit. Not that you have to fast to regulate your blood insulin, but it does do that. Yeah. I'm thinking about trying to market a class for Cal Poly that I would be interested in maybe teaching, or one of us should, like a general topical nutrition class. It's like 100 level, meet once a week, open it up to everybody. Everybody comes in there and I'm like, okay, intermittent fasting today, folks. Right? And everybody goes, hey, let's go. And then they'll walk out like, you said you probably shouldn't do it. I didn't know what he's talking about. Okay, that's fine. Hey, ketogenic diets today, right? We'll talk about that in a minute. What else? All kinds of stuff like that I think might be interesting. And then maybe we could make it so that it could be an elective for nutrition students. Maybe make it a 1 unit. But then everybody else could come.
Yeah.
And just go. We talk about obesity. We could talk about type 2 diabetes. We could talk about exercise and nutrition. Stuff like that. I'm trying to. I just think I'm tempted, kind of, to look for more work and set up that class. But I think that'd be interesting because everybody eats, right? So now let's talk about HDL. Why are they considered good? Here's their function. They do what's called reverse cholesterol transport. In other words, they're taking cholesterol from extrahepatic tissues to the liver. And so that's why when you see it in the blood, you say, hey, that's a good cholesterol. That's going in a good place, probably out of the body. And one of the things about HDL is ApoA1 is crucial for HDL structure and also activates an enzyme called LCAP. We'll look at that in a second. And cholesterol ester and HDL can be transported to the liver for excretion. So that's what it's doing. And here's a diagram I took from another textbook because it shows a visual of what's happening here. And so I mentioned before that HDL the new HDL are made in the liver and intestinal cells. And they go into the blood as these kind of, naive small particles. They go into the blood, and initially they are disc-like. And the reason they're like discs is because of this. Like, let's say this is an HDL. It's kind of flat. And this just continues on, right? And then you have the other bilayer that's here. I won't draw all this. They come out basically not full of anything. So they're just kind of like flat discs. They go into the blood. They're very small, very dense. They go into the blood, and then they can go in between the endothelial cells. That's what these are. They go in between the endothelial cells into the underlying tissue, and then they interact with the surface of the cells. Remember those cells have cholesterol in their membranes? If they have a lot of cholesterol, HDL can pull it from the membrane. So you have internal regulation of cholesterol values, but you also have LDL that can pull some cholesterol off the cell surface. And that's what's going on here. So FC means free cholesterol. In other words, it's not cholesterol ester. All right, so this HDL particle is scavenging cholesterol from cells. And then look what it's doing. It's getting bigger. It's picking up cholesterol, but initially what it's doing is, maybe this will work. It's putting the cholesterol on its surface. So it's getting bigger, but it's not really a sphere yet. Still kind of flat. And then it goes this way, comes back out. Still a disc. But it's got A1 here. Here's ApoA1. Yes, Brenda?
Is the cholesterol being pulled from inside the cell or from the membrane?
Typically from the membrane.
So as it goes through the whole.
Yeah, the HDL aren't going inside the cell, they're pulling it from the surface. Yeah. Oh, and it's picking up phospholipids too. That's how it gets bigger. Phospholipids and cholesterol. Now it goes back into the blood. Gets to a certain size, goes back into the blood by the lymph and then into the blood. Right? It's basically in the lymph here. It's back into the blood. Now look at it, it's bigger. It also picks up this enzyme called lecithin cholesterol acetyltransferase. That's on the next slide. LCAP converts cholesterol to cholesterol ester. See the way it went from a disc? Now it's round. Why? Because the cholesterol moved from here to the inside. And I'll show you the reaction now, and then we'll go back to this. There's words that don't mean anything on the left side, and a picture on the right, which for me was like, oh, okay. So there's cholesterol, upper right corner. There's phosphatidylcholine, lecithin, right? Very, very common phospholipid in our body. Very common in lipoproteins, very common on the surface of cells, plasma membranes. Remember, lysophosphatidylcholine has two fatty acids and choline over on the right side of this. And so now look what happens. That's in pink because that fatty acid is removed from lecithin. That fatty acid is attached to cholesterol. Right? So now it's a cholesterol ester. And this is lysolecithin. It's had that fatty acid attached. So let's read this. It says HDL scavenge excess cholesterol from tissues. HDL contain ApoA1. Lecithin cholesterol acetyltransferase, as its name implies, just takes a fatty acid from lecithin, transfers it to cholesterol. That's what it does. And that forms phosphatidylcholine, transfers fatty acid from phosphatidylcholine to cholesterol to form cholesterol ester. And then the cholesterol ester is embedded in the core of the HDL. So now it does this. Now it goes inside. So now you would have basically your cholesterol ester molecule. Right, that's got your cholesterol and now a fatty acid attached. Why did it move? It no longer has that polar group. Right? And so when you start moving those from the surface to the inside, the shape of HDL changes. So now here's the point of it. And in your notes, you should have a sentence that's something like this. Moving cholesterol from the HDL surface to the inside of HDL increases the likelihood the cholesterol is transported to the liver rather than transferred to another lipoprotein in the blood. Rather than transferred to another lipoprotein in the blood. And that would most likely be what? What's the highest concentration? LDL. So the dirty little secret is cholesterol on the surface of lipoproteins can move from one particle to another. So let's say you have let's say I have an HDL here and it's got cholesterol on its surface. If it's close to an LDL particle, that cholesterol can transfer, right? Because they come in contact in the blood. And so now what happens? Now your cholesterol is on LDL, and you don't want that. You want this cholesterol to become a cholesterol ester. It won't transfer. So it increases the likelihood that this particle, this HDL, is actually going to take all of the cholesterol that it picked up and move it to the liver. That's why LCAT's important. And that's why cholesterol in HDL is considered to be beneficial. So the more HDL you have in the blood, the more cholesterol you can scavenge and move to the liver for secretion. Excretion, I should say. Good there? Okay. Maybe let's take our break here and then we'll do lipid lecture 4 in a minute.